Hello students, in the previous sessions we have gained enough background knowledge to understand Mendel's experiments. In my today's session we will see about the Mendel's laws of inheritance, law of dominance, law of segregation, law of independent assortment, and a few important type of crosses that are done in Mendel's experiments. Law of dominance. This is the first law of inheritance that we will see today. Statement of law of dominance. When two homozygous individuals with one or more sets of contrasting characters are crossed, the alleles, that is the characters that appear in the F1, that is the first generation, are dominant, and those which do not appear in the F1 generation are recessive. What does this statement mean? In a monohybrid and dihybrid crosses, the phenotypic characters are controlled by a discrete unit or factors. In a dissimilar pair of factors, one member of the pair dominates, that is dominant over the other, which becomes recessive. The law of dominance is used to explain the expression of only one of the parental characters of a monohybrid cross in F1 and the expression of both in F2. The best way to understand the law of dominance is to understand a monohybrid cross. Just to recall, a cross between parents differing in only one heritable trait is called a monohybrid cross. Example, a cross of pure tall and pure dwarf plants. Mendel's performed the monohybrid cross between two pea plants with only one pair of contrasting characters. Have a look at this diagrammatic representation here. The first generation, that is the parental generation, you have a tall plant and a dwarf plant. Now the tall character is denoted with capital letters and the dwarf character is denoted with small letters. If you remember, in the previous sessions I had explained that the dominant character, that is the character which appears in the F1 generation, is denoted with a capital letter and the recessive character is denoted with a small letter. So this is your parental generation. When they are crossed, you have your first generation, that is F1. Both these gametes come together and you have one capital T, one small t coming together. So all the genotypes are capital T, small t, and phenotype, all of them are tall. If you put this in a Punnett square here, you have the first parent and the second parent. You put them across the squares and try to bring all the expected alleles together. All the F1 plants are heterozygous and they all have a tall stem. Then you further move with the cross, that is, you allow the selfing of your F1 generation. So both the parents are going to have the same genotype, and when you bring the gametes together, these are the possible genotypes of your F2 generation. If you put this in a Punnett square, that is one capital T, one small t from the paternal side, one capital T, one small t from the maternal side. If you bring together, you can see in one square you have two capital T's, one you have one capital, one small, same here, and two small. So if you have to put this into the ratios, the genotypic ratio is going to be 1 pure tall, 2 hybrid tall, and 1 pure dwarf. So the ratio comes to be 1:2:1. And the phenotype is going to be 3 tall and 1 dwarf, which is 3:1. So from this experiment, it's very easy to understand in the first generation itself that the characters, I have taken two different characters. I have a tall character and I have a dwarf character, and in the F1 only the tall character is seen. It means that the tall character is dominant over the dwarf character, which proves our law of dominance. The next law that we are about to see is the law of segregation. Statement of this law: the law states that when hybrid, that is the F1 generation, when it forms gametes, the alleles segregate from each other and enter in different gametes. The gametes formed are pure in that they carry only one allele each. Either it is going to be a dominant allele or a recessive allele. Hence, this law is also described as law of purity of gametes. Explanation about the statement. This law is based on the fact that the alleles do not show any blending, mixing, and both the alleles, that are the characters, are recovered as such in your second generation, that is the F2 generation, though one of these is not seen at the F1 stage. During formation of gametes, these two alleles, that is factors, obviously separate or they segregate. Otherwise, the recessive type will never appear in the F2 generation. The gametes which are formed are always pure for a particular character or a trait. A gamete may carry either the dominant or the recessive factor, but not both. That is why this law is called as law of purity of gamete. Let us go back to the monohybrid cross to understand the law of segregation better. See, have a look at the monohybrid cross again. We need to now highlight the F2 generation to understand the law of segregation. Here, when we are selfing our F1 hybrid, you can see the two alleles that are together, the capital T and the small t. They come together. Though in the F1 generation, the dominant character, that is the tallness, is visible, but the dwarfness does not disappear. It remains together and they separate out at the time of gamete formation. And since they do not mix and they separate, that's how the dwarf trait, which was not visible in the F1 generation, reappears in the F2 generation. And this particular factor proves the law. After the monohybrid cross, let us have a look at the dihybrid cross. A cross between parents differing in two hereditable traits is called as a dihybrid cross. Example, a cross of pure tall, round seed plant with a dwarf wrinkle seed plant. Mendel also performed dihybrid crosses between pea plants that differ in two pairs of contrasting characters. Here, you have a diagrammatic representation of a cross between parents differing in two pairs of contrasting character. One is the color of the seed and the other character is the shape of the seed, which is yellow round and green wrinkled. As you can see, the yellow round is a dominant trait and green wrinkled is the recessive trait. At the time of gamete formation, one Y, one R, and from here one small y and small r come together to give you your F1 generation, which is having both the characters, that is the dominant character, genes for the dominant character as well as the genes for the recessive character. But on the basis of law of dominance, your F1 hybrid is going to appear yellow round. When it is allowed to self for your next generation, you will have these different alleles forming different pairs. Here you can have a look. Here you have one capital Y, one capital R coming together in the gamete. Then you could have one capital Y and the other small r. Then you could have one small y and a small r, and the last combination is the small y and small r. And you have to put them across in a Punnett square. One is the paternal set of alleles and the other one is the maternal set of alleles. Then just bring together as you were doing in your monohybrid cross. When they'll all come together, you will see that you get around 9 yellow round seeds out of 16, green 3 out of 16 green round seeds, 3 out of 16 yellow round seeds, and 1 out of 16 green wrinkled seeds, which give you a phenotypic ratio of 9:3:3:1. Here I have also given you a solved example of the activity that is in your textbook. You can try it out and you can check it by pausing this video and verify whether you are successful in filling the grid. Now, when we see this, you can see that both the characters are getting inheritance independently. That is, inheritance of one character, that is the color, is independent of the shape of the seed. So when you consider two characters at time, one character the inheritance of one does not affect the inheritance of the another character. They are inheritant independent of one another, which this particular observation gives you, takes you to our next law, which is law of independent assortment. So the law of independent assortment, it states that when hybrid possessing two or more pairs of contrasting characters or factors, that is the alleles, form gametes, the factors in each pair segregate independently of the other pair. Explanation: this law is based on the dihybrid cross that I have explained to you already. It is a basic principle of genetics developed by Mendel. It describes how different genes or alleles present on separate chromosomes independently separate from each other during formation of gametes. These alleles are then randomly united in fertilization. In dihybrid cross, F2 phenotypic ratio is 9:3:3:1. It indicates that the two pairs of characters behave independent of each other. It can also be concluded that the two characters under considerations are assorted independently, giving rise to different combinations. If you have a look at the dihybrid cross, you will see along with the parental combination, you come across certain new combinations also, which is again a proof of the independent assortment. Okay, now we come to a next topic, that is how to find the unknown genotype. We have two ways of crosses, that is a back cross and a test cross. Back cross, the F1 individuals obtained in a cross are usually self to get the F2 progeny. They can also be crossed with one of the two parents from which they are derived, that is either the recessive or the dominant. Such a cross is called as a back cross. Test cross. The cross of F1 hybrid with the homozygous recessive parent is known as the test cross. Please note only the homozygous recessive parent. In a back cross, you can do either with the recessive or the dominant. It is used to test whether an individual is homozygous, that is pure, or it is heterozygous, hybrid. Test cross is easy, simple, repeatable and predictable. Test cross can be used to find out the genotype of any plant with dominant expressions, but it is not known whether it is homozygous, pure or heterozygous for that trait. Have a look at this diagrammatic representation here. I have a flower and I'm not aware of its genotype, whether it is having two capital W's or two small w's. I cross it back with the homozygous recessive parent here. Two small w's, two capital W's. All the plants, if are violet, then the unknown genotype is homozygous and is dominant. But if I cross it back with the recessive parent and I get a combination where I have half the flowers that are violet and half that are white, it will clearly tell me that the unknown flower is a heterozygous flower. That is, if the ratio comes out to be 1:1. If you cross it back with the dominant parent, at all times you are only going to get the dominant trait. So it will be difficult to identify whether the phenotype is a dominant character or that is whether it is homozygous or whether it is heterozygous. So the best way to do is to cross it back with its recessive parent. And that's why it is also said that a test cross is a back cross, but a back cross is not a test cross, because in back cross you use either the recessive or the dominant, but in the test cross, don't forget, we always use only the recessive parent. Thank you students for participating in this lecture. We will meet soon again with the next topic. Till then, take care, be safe and keep learning.